Good day, grade 11s. In this lesson today, we will be having a look at the anatomy of the human digestive system. The human digestive system consists of various organs required to perform the different processes involved in nutrition. These organs form part of the alimentary canal, which is a long muscular tube that extends from the mouth to the anus. Additional organs called accessory organs assist in the functioning of the digestive system but do not form part of the alimentary canal. The alimentary canal only consists of the mouth, the esophagus, the stomach, the small intestine, and the large intestine. The accessory organs are not part of the alimentary canal as food never passes through these organs. They only assist by either secreting various substances needed for digestion or receive nutrients only after it has been absorbed into the blood. These organs are the salivary glands, the liver, the gallbladder, and the pancreas. The first part of the alimentary canal we'll be focusing on is the mouth or the mouth cavity. The mouth cavity consists of the teeth, the tongue, the uvula, which closes the nasal passageway and prevents food from going into the nasal passageway, as well as the openings of the salivary glands, which is where salivary glands will secrete saliva into the mouth to aid with digestion. The function of the mouth cavity is digestion, both mechanical digestion, which is the physical breakdown of food by the teeth and the tongue into smaller food particles, as well as chemical digestion, which is the breakdown of those food particles into nutrients which can be dissolved into water so that it can later absorb into the blood. Chemical digestion is aided by water in the saliva as well as the enzymes which are secreted by the salivary glands. In the mouth, the food is chewed and mixed with saliva to form a ball of food called a bolus. From there, the food passes into the pharynx, which is the throat area. The pharynx is the area where the mouth cavity and the nasal passageways meet. The pharynx also leads to two openings lower down, the esophagus, which is where the food has to pass through to go down to the stomach, as well as the trachea, which is where air passes through to go to the lungs. We will learn more about the trachea later when we look at the respiratory system. To control food going down the esophagus and prevent it from going down the trachea, there's a cartilaginous flap called the epiglottis, which prevents food from going down the trachea. So the function of the pharynx is to control the passage of air into the trachea, and then importantly for the digestive system, to control the food going into the esophagus. The epiglottis, as mentioned, closes the opening to the trachea, called the glottis, during swallowing to prevent food from going down the trachea. Over here you can see an animation of the functioning of the epiglottis. So you can see the epiglottis there is the cartilaginous flap, and during swallowing it closes the trachea to prevent the food from entering the trachea and guides it down into the esophagus. This brings us then to the esophagus. The esophagus is a long muscular tube that is situated behind the trachea and connects the pharynx to the stomach. The function of the esophagus is to move the bolus of food from the pharynx to the stomach. This is achieved by the rhythmical contractions of the muscles of the esophagus pushing the bolus of food through the thin tube into the stomach. This rhythmical contraction of the muscles is referred to as peristalsis. Over here you can see an animation of how the esophagus pushes the food into the stomach. As mentioned, peristalsis is the rhythmical contraction of muscles to move a food bolus down the esophagus. This is achieved by alternating contractions and relaxations of the muscles of the esophagus. there are two sets of muscles in the esophagus, the circular muscles and the longitudinal muscles. The circular muscles are, as the name suggests, circular in their structure, meaning when they contract, they close or they reduce the size of the ring, and this then pushes the food down the esophagus. The longitudinal muscles run longitudinally along the esophagus, so when they contract, they further push the food down the esophagus. So by alternating the contractions and relaxations of the circular and longitudinal muscles, it pushes the food down the esophagus. From the esophagus, the food then moves into the stomach. The stomach is a thick muscular bag filled with gastric juices. The lining of the stomach is made of thick muscular walls that facilitate the churning movements required for digestion. The stomach also has two sphincter muscles that control the openings of the stomach. The cardiac sphincter controls the opening to the esophagus, while the pyloric sphincter controls the opening to the small intestine. The main function of the stomach is digestion. Mechanical digestion is achieved by churning movements of the muscles of the stomach that mixes and breaks up the food, while chemical digestion is achieved by the juices that is secreted by the stomach, called the gastric juices, which is going to facilitate the chemical digestion. The mixture of food and gastric juices inside of the stomach is referred to as chyme, and it is this which is going to pass on to the small intestine, where further digestion and absorption is going to take place. After food has been thoroughly broken down by the stomach, the contents of the stomach, called chyme, moves into the small intestine. The small intestine is a long muscular tube, about 7 meters long, where most of the absorption is going to take place. The small intestine consists of three regions: the duodenum, which is the first part of the small intestine and connects to the stomach; the jejunum, which is the middle part of the small intestine; and the ileum, which is the last part of the small intestine and connects with the large intestine. As mentioned, the main function of the small intestine is absorption. However, some digestion still takes place in the first part of the small intestine, the duodenum, by means of some digestive juices which is secreted and added to the duodenum by some of the accessory organs. The main function of the small intestine is absorption, and we will look more at this process a bit later on. To move the chyme through the very long small intestine, rhythmical contractions of the muscles of the small intestine achieves this movement by means of peristalsis. After absorption of nutrients has taken place in the small intestine, the contents then move on to the large intestine. The large intestine also consists of various regions. The first region of the large intestine is called the cecum. The cecum is the area of the large intestine that connects with the small intestine and receives the contents from the small intestine. Connected to the cecum is an appendage that we call the appendix. You might have heard about the appendix before, as this part of the large intestine oftentimes becomes inflamed and has to be removed as a result. The exact function of the appendix is not well known and is still being debated amongst scientists, so the function of the appendix is not something that you have to know. From there, the contents then move on to the colon. The colon consists of three regions: the ascending colon, which means going up; the transverse colon, which means this going from side to side; and the descending colon, which is the part of the colon that goes downwards. From there, it then moves into the rectum, and finally, the expulsion of waste, called egestion, is controlled by a sphincter muscle called the anus. So the function of the large intestine is mainly to absorb water and any other useful substances like vitamins that wasn't absorbed by the small intestine. Since the large intestine absorbs a lot of water, the contents of the large intestine becomes dried out as it goes along the large intestine. And as a result, to help lubricate the movement of this undigested waste through the colon, mucus is secreted to allow the contents to move through more easily. Then lastly, any undigested waste that must be egested is stored in the rectum until a point where it can be egested. The large intestine is the last part of the alimentary canal. So that then moves us on to the additional organs that assist the functioning of the digestive system, called the accessory organs. The first accessory organ we're going to look at is the salivary glands. The salivary glands are three pairs of salivary glands that secrete saliva into the mouth cavity via ducts that connect or open into the mouth cavity. You do not, however, need to know the names of the three pairs of salivary glands. The function of the salivary glands is to secrete saliva containing enzymes and water that assist the chemical digestion that takes place inside of the mouth cavity. The next accessory organ we're going to be looking at is the liver. The liver is the largest gland in the body, and it is situated below the diaphragm. It consists of two lobes, a larger right-hand lobe and a smaller left-hand lobe. The liver has many important functions in the body. One of the important functions that relate to the digestive system is the production of bile. Bile is a pigment that is created by the liver that assists with the breakdown of fats in the body. We will look at the functioning of bile a bit later on. Bile is then stored in the gallbladder, which we will also look at a bit later. Another important function that relates to the digestive system is the conversion of glucose to glycogen. Glucose is an important nutrient in the body. However, high amounts of glucose in the blood is very dangerous to humans. As a result, this glucose has to be converted to glycogen so it can be stored for later use. The liver also breaks down excess amino acids. It breaks the amino acids into urea and glucose, and the process of breaking down amino acids is called deamination. Another important function of the liver is to detoxify toxins like alcohol and store vitamins. As mentioned, bile being produced in the liver is stored in an organ called the gallbladder. The gallbladder is a muscular bag that is situated just below the liver. The function of the gallbladder is to store and release bile when it is required. It releases the bile into the duodenum via the bile duct, where the breakdown of fats will then take place. So as mentioned, the function of bile is to assist with fat digestion. The next accessory organ we're going to look at is the pancreas. The pancreas is a large gland that is situated below the stomach. The pancreas consists of two types of cells: normal pancreatic cells as well as cells of the islets of Langerhans. The function of the pancreas for the digestive system is twofold. The normal pancreatic cells produce pancreatic juice, which contains enzymes which assist with digestion. We will look at the pancreatic enzymes a bit later on when we look at the digestion process more in detail. The second function of the pancreas is achieved by the cells of the islets of Langerhans. These cells produce hormones called insulin and glucagon, which regulates the blood glucose levels. If the blood glucose levels goes too high, the hormone insulin decreases the blood glucose levels, and if the blood glucose levels goes too low, the hormone glucagon increases the blood glucose levels again. We will look at this functioning of the regulation of blood glucose levels a bit later on. So if we look at the digestive system as a whole, we should be able to identify the following organs and know their functions. The first area is the mouth or the mouth cavity, where mechanical and chemical digestion will start. Chemical digestion in the mouth is assisted by the salivary glands, which secrete saliva into the mouth cavity. The mixture of food and saliva in the mouth forms a ball of food called the bolus, which is then transported to the pharynx, where it will then be transported to the esophagus, which will push the bolus of food down into the stomach by peristaltic movements. In the stomach, mechanical and chemical digestion continues to turn that food into a gray sludge that we call chyme. The chyme then moves into the small intestine. The first part of the small intestine is called the duodenum, where some digestion still takes place. Then into the jejunum, and then into the ileum, which forms the three parts of the small intestine. From there, the contents is then moved onto the large intestine, specifically the ascending colon, the appendix, the transverse colon, and the descending colon, which forms the large intestine. From there, the undigested remains then move into the rectum, where it is stored until it can be egested, assisted by the functioning of the sphincter muscle called the anus. The accessory organs that assist the functioning of the digestive system is the liver, the pancreas, the gallbladder, which secretes bile into the duodenum via the bile duct. Now that we have a good understanding of the anatomy of the human digestive system, we can start looking at how does the digestive system function. The functioning of the human digestive system is divided into 5 processes. These 5 processes are ingestion, which is where food enters the alimentary canal through the mouth. Then digestion, which is the mechanical and chemical breakdown of food. Followed by absorption, where the food that has been broken down into nutrients can be absorbed into the blood. Followed by assimilation, which is where the cells then use these nutrients to build new cells. And then finally, egestion, where any undigested remains that we call feces is removed from the body. The first process of the digestive system that we will be focusing on is digestion, as ingestion doesn't really require any further analysis. So digestion is divided into 2 parts. There's mechanical digestion and chemical digestion. Mechanical digestion is the physical breakdown of food. This is where food is broken down into smaller parts by the mouth and the stomach, as to expose more surface area for the enzymes to break the food down even further. So the purpose of mechanical digestion is to break it down into small particles to increase the surface area for chemical digestion later on. So chemical digestion is the breakdown of food particles into nutrients so that the nutrients can dissolve in water and be absorbed in the small intestine. This chemical breakdown of food is called hydrolysis. Hydrolysis is a chemical reaction where a molecule is broken down by means of water, and in digestion it is assisted by the functioning of enzymes. We will look more in depth at how different enzymes break down different food molecules. As you know, foods are divided into 3 different categories: carbohydrates, proteins, and lipids. This is because the chemical structure of these foods are different, and as a result, they are broken down differently by our bodies. So we will be looking at the chemical digestion of these 3 different food groups individually. First, we will be looking at carbohydrates. Carbohydrates are broken down by a group of enzymes called carbohydrase. These enzymes are different enzymes that we all group together as carbohydrase. They are secreted in various parts of the body. They are secreted by the mouth, the small intestine, and the pancreas. They function within the mouth and the small intestine. So the pancreas will secrete enzymes which will then be added to the duodenum of the small intestine, where they will then break down the food. As you know, enzymes work best at a very narrow pH level. So as a result, the pH levels within these different areas of the body needs to be controlled within those very narrow margins as to ensure the optimal functioning of these enzymes. The optimal pH for the carbohydrates in the mouth is an alkaline pH, whereas the optimal pH for the enzymes within the small intestine is a neutral pH. The end product of all of this chemical digestion of carbohydrates is glucose, which is a nutrient that can be absorbed into the blood and is required by our cells for processes like respiration. The second food group are proteins, and proteins are broken down by a group of enzymes called protease. These enzymes are secreted by the stomach, the small intestine, and the pancreas, and they function within the stomach and small intestine. So once again, the enzymes created by the pancreas are added to the duodenum of the small intestine via a duct, where they will then break down the proteins within the small intestine. The optimal pH levels for these enzymes is acidic in the stomach and neutral in the small intestine. The end product of all of this chemical digestion of proteins is amino acids, which our body requires to build various proteins that our body needs. The last food group is lipids. Lipids is the fats and oils within our diet. The enzymes that breaks down lipids is lipase and is secreted by the small intestine and also functions within the small intestine. The pH level at which lipids functions best is at a neutral pH. So as you can see, all of the enzymes that function within the small intestine functions best at a neutral pH, which means the small intestine always has to be kept at a neutral pH. The end product of the digestion of lipids is fatty acids and glycerol. An important thing to note about the breakdown of lipids is the functioning of bile to assist the breakdown of lipids. Fats or lipids are hydrophobic, which means they do not mix with water. This makes it very difficult for the enzymes to get access to the fats to break them down. As a result, bile emulsifies the fats, which means bile breaks the fat down into smaller droplets so that the enzymes can get access to the lipids and break it down into fatty acids. The next process of the digestive system is absorption. After digestion has taken place, the food particles should be broken down into their simplest forms, nutrients. These nutrients then dissolve into water and can be absorbed into the bloodstream at the small and large intestine. Most absorption, however, takes place in the small intestine, and the small intestine is specifically adapted to maximize absorption. In order to better understand the process of absorption, we need to have a look at the structure of the small intestine, as the small intestine is specifically adapted for absorption. If we have a look through the cross-section of the small intestine, we will find a number of layers. The first layer is the outer protective layer of the small intestine called the serosa. This is followed by two muscle layers. The first muscle layer is longitudinal muscles, which run along the small intestine, followed by a layer of circular muscles, which forms rings around the small intestine, and these two muscles together facilitate the peristaltic movements which move the chyme through the small intestine. This is then followed by two layers of mucosal layers, or two mucosal layers. First the submucosa and then the mucosa. The mucosa is folded to increase the surface area inside of the small intestine for increased area for absorption. The mucosa is then also has structures on them called villi, which further increase the surface area for absorption. The inside of the small intestine is referred to as the lumen. So we've seen that the small intestine is specifically adapted to maximize absorption. We saw that the small intestine is long, plus minus 7 meters long, which ensures that the food remains inside of the small intestine for a long period of time to maximize the time for absorption. We also saw that the mucosa is folded, which increases the surface area for absorption, and similarly it has structures on the mucosal layer called villi, which further increases the surface area, and those villi are lined with epithelial cells which have microvilli, which further increase the area for absorption. So all of this ensures that all the nutrients, or the maximum amount of nutrients, are absorbed while inside of the small intestine. So we've now looked at the cross-section of the small intestine to see how the small intestine is adapted to maximize absorption, and we said it has folds. The mucosal layers are folded to increase the surface area, and then that these folds has even smaller structures on it to further increase the surface area, and these structures are called villi. So we will now look at how these villi, or a singular is called the villus, how a villus is adapted to further increase the surface area for absorption. Zooming in and looking at the structure of a villus, we can start to see how absorption will actually take place within the small intestine. The first thing that we'll notice is that the outer lining of the villus, which means the innermost lining of the small intestine, is a layer of columnar epithelial cells. This is a single layer of cells, which means diffusion can take place very easily and very quickly, as there's only one layer of cells that nutrients needs to travel through. We'll also notice that these columnar epithelial cells, which are roughly rectangular shaped, have a layer of hair-like structures on them called microvilli. These microvilli further increases the surface area for absorption. We'll also notice that some of the columnar epithelial cells look slightly different. These are called goblet cells. And goblet cells produce mucus, which forms a mucus lining on the inside of the small intestine, where nutrients can dissolve into so that they can diffuse into the villus at a quicker rate. Then we'll see that there's a rich blood supply that supplies the villus. An arteriole brings blood to the villus, and it then divides off into many capillaries, forming a capillary network, which then join up again to form a venule, which will take blood away from the villus. This means nutrients that dissolves in the mucus can then be diffused into the villus and diffuse into the blood vessels, which will then be taken away by the venule and be taken into the bloodstream. We'll also notice that there's another structure here called the lacteal. This vessel is part of the lymphatic system, and this is where fats will absorb into. So most nutrients diffuse into the blood vessels and is taken to the bloodstream. However, fats gets absorbed into the lacteal, which gets taken to the lymphatic system. So after having looked at the structure of the villus, we can understand how the villus and the small intestine is adapted for efficient absorption of nutrients into the blood. We mentioned that the villus has one layer of columnar epithelial cells for fast and easy diffusion of nutrients into the blood. We saw that there were many blood capillaries within the villus so that nutrients can be absorbed into blood efficiently and quickly. We saw that the villus had a lacteal, which was responsible for the absorption or which is responsible for the absorption of fats. We saw that the columnar epithelial cells have goblet cells amongst them to secrete mucus so that the nutrients can dissolve in a layer of mucus and be absorbed into the blood more efficiently. We saw that the columnar epithelial cells has microvilli to further increase the surface area for absorption. And something that we weren't able to see, but it is another adaptation of the villus, is the many mitochondria of the cells of the villus, which produces energy required for active transport of nutrients into the blood vessels of the villus. Now that we've had a look at the structure of the small intestine and the villus, in order to understand how these structures facilitate absorption, we can look at the process of absorption in a little more detail. We've said that the end products of digestion gets absorbed by the villi of the small intestine. These end products or nutrients are glucose, amino acids, fatty acids, and glycerol, as well as some other substances like vitamins, minerals, and water. All these substances are absorbed into the small intestine in different ways. Some of them require energy as they have to be transported against the concentration gradient, meaning from a low concentration to a high concentration. This type of transport is referred to as active transport. Other substances do not require energy as they go along the concentration gradient, meaning from a high concentration to a low concentration. This type of movement is called passive transport. It is also worth noting that not all substances are absorbed by the small intestine, as a large amount of water and certain vitamins and minerals are also absorbed by the colon. We will look at how each different substance is absorbed either by the small intestine or the large intestine. In order to differentiate how different nutrients are absorbed by the small and large intestine, we've placed the nutrients into a table to easily see how each nutrient is absorbed. Glucose is the end product of carbohydrates and is actively absorbed. This means it requires energy. It is absorbed into the blood capillaries of the villus. Similarly, amino acids, the end products of proteins, are actively absorbed into the blood capillaries of the villus. Lipids, however, which are broken down into glycerol and fatty acids, are passively absorbed, meaning no energy is required, by the process of diffusion into the lacteals. Other substances like vitamins, minerals, and water are also absorbed in a variety of different ways. Vitamins can be divided into fat-soluble vitamins, meaning it dissolves in fat, and water-soluble vitamins, meaning vitamins that dissolve in water. Fat-soluble vitamins are passively absorbed into the capillaries. Water-soluble vitamins are also passively absorbed, but require the aid of carrier molecules to transport them into the capillaries. Minerals can either be passively or actively absorbed into the capillaries, depending on the concentration of minerals in the blood. Water, however, is also passively absorbed by diffusion into the capillaries, and as mentioned, a large amount of water is absorbed at the large intestine as opposed to the small intestine. Once the nutrients have been absorbed, they can be transported to the cells where they are needed for various life processes. Different nutrients, however, take different paths in getting to the cells. Nutrients like glucose and amino acids that got absorbed by the blood capillaries can go directly into the blood system. However, they first need to make a stop by the liver. We will look at this process in a second. Fatty acids and glycerol that got absorbed by the lacteals enters the lymphatic system. The lymphatic system will eventually also enter the bloodstream. As mentioned, the nutrients that enter the capillaries first need to make a pit stop at the liver. This is facilitated by the hepatic portal system, which takes all the nutrients that get absorbed from the small and the large intestine to the liver. The hepatic portal system is a network of blood vessels that run from the stomach, spleen, pancreas, small intestine, and large intestine, and is transported via the hepatic portal vein to the liver, where the liver has a couple of functions before adding that blood to the rest of the circulatory system to be delivered to the cells. We will look at a couple of the functions that the liver performs in the hepatic portal system next. The purpose of the liver in the hepatic portal system that relates to the digestive system is twofold. Firstly, it converts excess glucose into glycogen to store for later, as an excess amount of glucose in the blood is problematic for humans. Secondly, it converts excess amino acids into urea and glucose through the process of deamination. Urea, however, is a waste product that needs to be excreted, so the urea will be transported to the kidneys to be excreted later. The remainder of the nutrients is added to the blood and then is transported to the rest of the body. Once nutrients enter the blood system, they can be taken up by cells to use for various metabolic activities as well as to build new cellular structures. This process is called assimilation. Examples of uses of nutrients by cells are glucose that is used for respiration to make energy, or amino acids which are used to build new proteins needed throughout the body, and fatty acids and glycerol are used to make phospholipids which are needed to make and repair cell membranes. The last process of the digestive system is egestion. Egestion is the process whereby any unabsorbed semi-solid waste that passes through the large intestine is expelled out of the body via the anus. As mentioned, the large intestine is responsible for absorbing a large amount of water and minerals, which turns the semi-solid waste more solid as it moves through the large intestine. The last aspect of human nutrition that we're going to talk about is homeostasis and a homeostatic control of blood glucose levels. But before we can dive into that, we must first understand what is meant by homeostasis. Homeostasis is a broad concept referring to all the ways in which an organism maintains a constant internal environment. It is necessary that organisms maintain a constant internal environment, as the systems and processes that keep them alive only function within a narrow range of conditions. So the definition for homeostasis that we use is the maintenance of a constant internal environment. So then why is it necessary that organisms maintain constant blood glucose levels? Well, firstly, we need to know what is blood glucose levels. Blood glucose levels is simply the amount of glucose in the blood. And this typically increases after we eat as the nutrients are broken down and the glucose is absorbed into the blood. But you might think to yourself, glucose is a vital nutrient that is needed by organisms for respiration. However, if the amount of glucose in the blood is elevated for too long, it can cause damage to the blood vessels, and this can then lead to further cardiovascular problems down the road. As a result, it is vital that organisms, including humans, regulate their blood glucose levels. Organisms achieve this regulation through the secretion of two hormones, insulin and glucagon, and that's what we will be looking at in understanding how these two hormones manage our blood glucose levels. So we will look at what happens when there's an elevated blood glucose level as well as when there's a very low blood glucose level, and how these hormones bring our blood glucose levels back to a normal level. Firstly, we will look at the effect of a high blood glucose. When the glucose in the blood rises above normal, the beta cells of the islets of Langerhans of the pancreas detect this change in blood glucose. As a result, the beta cells will secrete a hormone called insulin into the blood. The blood will then travel via the circulatory system to various parts of the body. It will decrease the blood glucose in two ways. Firstly, it will cause normal body cells to absorb glucose into the cells and store that glucose till later. Secondly, it will stimulate the liver and the muscles to convert glucose into glycogen. Glycogen is a polysaccharide, which is a form in which animals store glucose, similarly to how plants store glucose in the form of starch. So both of these effects will decrease the blood glucose levels. And this decrease in blood glucose levels will be detected by the beta cells, which will in turn inhibit the secretion of insulin and will stop the decrease in blood glucose when the blood glucose reaches a normal level once again. We refer to this as a negative feedback mechanism. When the glucose levels in the blood drops below normal, a different hormone is secreted. So if the glucose concentration in the blood falls below normal, the alpha cells in the islets of Langerhans of the pancreas detects this change. The alpha cells will then secrete a hormone called glucagon. Glucagon increases the blood glucose levels. It does this by converting the glycogen in the muscle and the liver back into glucose and adding that glucose into the blood to increase the blood glucose levels. So the glucose concentration in the blood then increases, and this increase is then detected by the alpha cells, which in turn inhibit the secretion of glucagon and stops the increase in blood glucose as the blood glucose levels return to normal. Once again, this mechanism is referred to as a negative feedback mechanism. So let's look at the control of blood glucose in a bit of a different way and represent it graphically in the form of a flow diagram. So firstly, if the blood glucose levels increases above normal, it stimulates the beta cells of the islets of Langerhans of the pancreas to secrete a hormone called insulin. Insulin will then have two effects. It will stimulate the conversion of glucose to glycogen in the muscles and the liver, and it will stimulate the absorption of glucose in our body cells. Both of these will cause the glucose levels to return back to normal, which will be detected by the beta cells, which will inhibit the secretion of insulin, meaning the homeostasis has been restored. If the levels of blood glucose drops below normal, the alpha cells of the islets of Langerhans of the pancreas secrete a hormone called glucagon. Glucagon stimulates the conversion of glycogen to glucose in the muscles and the liver. This glucose is then released back into the blood, which increases the blood glucose levels, which means the blood glucose levels return to normal and the alpha cells inhibit the secretion of glucagon and homeostasis is restored. And this is the homeostatic control of blood glucose. So what happens if homeostasis of blood glucose levels cannot be achieved? Well, this oftentimes results in a condition called diabetes mellitus. You might have heard about diabetes before, as it is a very prevalent disease in our society today. Diabetes is a metabolic disease characterized by a chronic high blood glucose level. There are two types of diabetes. Type 1 diabetes is caused by the inability of beta cells to produce insulin, meaning not enough insulin enters the blood to reduce the blood glucose levels. Whereas type 2 diabetes is characterized by our cells being resistant to insulin, meaning our cells can no longer take up the insulin in order to reduce the blood glucose levels. Both of these result in the blood glucose being very high after meals and causing damage to our blood vessels and can result in various cardiovascular problems down the road. And that brings us to the end of this lesson on nutrition. I hope it was useful. There was a lot of things that was not discussed, but this should give you a good overview of the whole topic of nutrition, specifically relating to the human digestive system. Good luck.